Welcome to our last webinar of 2021 for the Knowledge Portal Network. And I'm happy to have you all here, see friendly faces and see some new faces. So today, I'm excited because we get to talk about a new research product from the team. And before I introduce our speakers, I actually want to give you a little bit of background on the motivation and the rationale for this. Just to begin, you all know us for the knowledge portals for common disease that we've built. Culminating in the Common Metabolic Disease Knowledge Portal that houses 5 disease-specific portals. In addition, the 3 other complex disease portals, including ALS, musculoskeletal disease, and lung disease. And these are built all on this very large data and software platform that we've built for several years now, which takes in genetic data from public and private investigators, integrates them with functional genomics data that are relevant to those diseases, and then runs them through a set of bioinformatic approaches and tools that are the best in the field that we've integrated and implemented in the resource, and then has a set of access mechanisms. So this is a very large platform that all integrated is the huge AMP or human genetics amplifier resource that powers all of these portals. And this is a larger resource, and it has a set of standard visualizations and representation that we have a very large team that builds, and we have production releases at a quarterly cadence. And it has, it says visualizations are very useful for the types of things it does. But however, in many cases, that heavyweight activity isn't necessarily what you need to fast track access to certain results. And so we found along the way that there was a need for representation of some novel approaches to make them accessible really fast for publication, or approaches that weren't standard to the genetic analysis representation that we have in our portal. They were clustering analysis, or they were distilled representations, or they were unique visualizations that didn't require the heavyweight, huge AMP portal. So for that, we found, for example, a couple of years ago, the example we worked with Miriam Udler, where there's genetic data and genomic data that are kind of unique, where she did some soft clustering analysis of type 2 diabetes loci across multiple traits. And she had these figures that included some heat maps and also some tables that you wanted to allow a user to drill down into the variants that contribute to those particular clusters. And your static figures didn't do justice to that. Moreover, as our field has progressed with genetic association studies, people are producing these integrative lists, distilled lists, if you will, of genes or effector transcripts with supportive evidence that allows a user who's not an expert to get the headline findings about a particular gene. These tables, you need to make them interactive. In a paper, they don't necessarily sing or resonate if they're a static figure. Moreover, there are these elegant visualizations that are people making to represent genetic and genomic annotations and results that you want to make interactive, and papers don't do justice to that. So all that said, we looked at our platform and said, we need a new arm, a new way to make these results accessible, fast, easy, and to adapt to the changing visualizations that people want to represent the results. So with that, DK Zhang on my team developed a brand new service, which we are calling BYOR, or Bring Your Own Results, portal service, which allows you to build your own portal. So we're going to talk about that today and some of the guiding principles behind that. So some of the things we've done so far is they're currently accessible on this AMP KP, several of them, in our KP Labs page, which is really the impetus for what we started doing this. And they include things that I was mentioning before, like the type 2 diabetes effector transcripts, where you want an interactive table where it was contributed by an investigator, and they wanted the results visualized the way that they intended, because that's the best way to represent the knowledge. But you want it integrated with the resource and the data that's underlining in the knowledge portal. So this is an example of something we built in that space for the type 2 diabetes transcripts, where we built a novel visualization to summarize the results and make them accessible to you. We also built standalone portals, which Maria's going to talk about a little more, where this portal was built for the APOL1 project we did with Matt Sampson and team, where you have a set of visualizations that aren't in the portal, that aren't on the knowledge portal page, but they're a set of tables and interactive results that are its own portal by itself. So you have differential expression, enrichment analysis, and those static visualizations from the paper became interactive on the portal. So with that, some guiding principles of how we build this. Essentially, it boils down to this. We want to allow you to have your results presented your way. We have a set of core capabilities that are essentially fundamental to genetic and genomic studies, the types of regression analysis and visualizations that are used in many of these studies that are accessible to you to build your own, bring your own results too. We want to collaborate to construct. We like to work with you. However, though, it's easy to use, as DK will demonstrate, that you can update your data as you go. If there's a new figure that has new data to it, you can update this yourself and do it yourself, essentially. Finally, you can stage access as well, which means if you want to just share them for yourself, visualize your own results, or share them with a set of collaborators, or make them accessible to the world is an easy way to stage that access so that that can be done for you. And finally, something that I really love, it's a matter of leverage. For those of you who are familiar with the Pirates of the Caribbean, what I like about this is these portals can be their own entity, but they can also take advantage of the types of data that are in the huge AMP resource. For example, in one collaboration, we're working with the Giant Consortium to represent their results across a set of traits for obesity, height, and what they want is to see their own results to compare and contrast their own genetics results, but they want to layer it with annotations. They don't have those annotations accessible to them, but we do. So we're hoping that we can allow these different types of features and different types of results people want to make accessible can also be integrated with the types of results and methods and approaches we have in the huge AMP resource to make it an even richer resource to the community. So with that, I'll turn it over to DK, Maria, and Kenny, who actually was wonderful to agree to come talk to us about a portal he built using this framework. So with that, I will stop sharing and turn it over to DK.
All right, this is DK Jing. I do take care of UI and UX on the in the knowledge portal team, and I'm the main one working on the research portal at this point. So by the way, the research portal service is in beta stage. We are keep developing. I'm adding new functionalities and the new visualizers. So yeah. So idea is pretty simple that we have 3 parts in this structure platform, if you call. So we have the configuration part and then presentation part and the data part. The data part can be same to configuration part, or it can be anywhere. If the data has like public access, then it can be, and the data is formatted in a way that the research portal can consume, then there wouldn't be any limitations. So in the configuration part, you can set security or data formatting or data visualization, and then from the presentation side, it sends signal to I send message to the configuration part with authentication. So as Noel mentioned, we can set this presentation part in 3 stages. One is the configuration is created but not opened. It's not accessible. And second part is it's opened only for the development purpose or review purpose. And third one is just an open public. So that is to protect your data and the research from being hacked on the way to present. And then, as I mentioned, and also it comes with a data table that has built-in sort capability, even for the locus. So probably every one of you know that Excel or any this spreadsheet tools, they don't sort locus pretty well. So it has this locus sort capability, and then it comes with filtering system that is auto-generated from the data based on your configuration, and it comes with 6 different visualizers. Depends on your data. You can choose and configure. And at this point, we are working on a new visualizer package, which will be actually used in our knowledge portal as well. Yeah, that's it. So I think you will get much better idea when actually Maria demos how you upload the data and how things are created on the presentation side. So I will stop sharing and pass it to Maria.
All right, thanks, DK. Let me share my screen. Okay. All right, so when we were planning this webinar, DK suggested that I should just create a table, upload a file and create a table live during the webinar. And I was a little hesitant, but actually it is so easy that I'm going to do that. And I'm speaking as somebody with basically no computational or programming expertise whatsoever. So here's the example. So we're working with this group from the Genetics of Osteoarthritis Consortium, who just published this paper with a huge new GWAS on osteoarthritis associations. And as part of the study, they predicted osteoarthritis effector genes. And there's a whole list and table of them in the paper, and they have various supporting evidence. And so we're working with them to create this table on the musculoskeletal knowledge portal. And so as an example, I've taken part of their data just to upload today. So here's, I'm showing you the comma separated values file. So this is simplified. It doesn't have any of the supporting evidence, but it's got the predicted effector genes and some other data points. And this is a comma separated values file. And so really it can contain any type of data, and it doesn't have to be an effector list. Obviously, it can be really any kind of biomedical data. So here's this CSV file. And so I'm going to go to the research portal interface to add content, and I'm going to add a research page. So a page is basically one data table, one data file, which creates an interactive data table, and it can also, the data can also be visualized in various ways that I'll show you in a minute. And then when we talk about a research portal, we mean multiple pages like this that can be linked together with navigation menus and so forth. So let's create this. Well, you can see from my browser that I actually did practice this. But okay, so we'll give it a title, and the page ID just has to be something unique. Now for the data points, you want to put the name of the data file. And so I'm going to go to the file manager here. And I already, let's see, the Zoom menu is getting in the way of my browser menu. So I've already uploaded this file, but the upload is super easy. It's just you click to upload and then you navigate to your file, just like uploading anything to the web. So I've uploaded this file, and I'm going to copy the name of it. Okay, and then go back to this interface and just paste the name in. And I'm not going to go over all these other fields. These are ways to configure the data and so give the table a legend, set filters and so forth. I'll point these things out when we look at the interface, but I'm not going to do them live. And here's some places to specify the visualizer that you want to use. I'll set the page mode to public. Oh yeah, and let me go back up to the top and copy the page ID because I'll need that to visualize the page. Okay, and then I'm going to save. Okay, and now I should be able to add that page ID to this URL. Yes, okay. So it was really that easy. And these tables are filterable. Sortable. The columns are sortable, and they're also filterable. I didn't add any filters to this table, but you can add custom filters to filter by any of the criteria in the main columns of your table. So let me show you the. So this is the table, the more complete osteoarthritis effector genes table that I'm working on with this group. And in this case, we've added the supporting evidence. And so this is an example of adding subtables. So for each row, we can expand this to see the supporting evidence for this prediction of this effector gene. So, and these evidence types are separated by whether they're genetic evidence, regulatory evidence, or phenotypic evidence. And so I was actually able to create these subtables myself with a little help from DK and with a great tutorial that he's working on. So it's really not too complicated. And here's an example of where there are different filters. So you can filter the table by gene name or score or the variant that is the lead, excuse me, the lead signal for this locus. Also, I should mention that you can add a research method. This tab is empty right now here, but you can add text and figures explaining what you've done in this visualization or in this table. Okay, I see we have some questions in the chat. Is there anything that's, okay, it's just Noel giving us some information. Okay, let's see. So yeah, as Noel mentioned, we've used this framework to build some visualizations that you have seen already in the portal. And if, for example, in the KP labs, we have the genetic loci clustering interface, which has the type of expandable table that we're talking about, and it also has the heat map. That is one of the visualizations that's easily available through this service. We also have lots of effector gene lists. I'll just show one of them. This is the list of predicted effector genes for coronary artery disease from the Cardiogram plus C4D consortium. And so here's a table like the one I showed you. I forgot to mention that in the table, you can also link gene names and variant names to their gene or variant pages in the portal if you wish. And this again has expandable evidence rows. Also, with this table, now there's a visualizer. This is one of the available visualizers, which is a score plot. So it's a Manhattan-like plot in that it lays out the signals along the chromosomes, but then it shows the number of predictors. So in this study, for each locus, the authors looked at 8 different predictors to classify genes as as predicted effectors. And so the y-axis shows the number of predictors that were satisfied by each of these genes or loci. All right, so let's see what we have. So here's an example of a really more complicated research portal, and you'll see another one from Kenny in a few minutes. So Noel mentioned this. This is a portal on a rare kidney disease that was created in collaboration with Matt Sampson's group at the Boston Children's, and it accompanies a manuscript they submitted on this. So as we said, you can add menus. You can have a multi-page portal and add menus to navigate between the pages. The pages can, of course, have just text. They can also have images. So like in this case, they created a very detailed tutorial to explain how to use and understand all of the visualizations on this portal. They've also got a downloads page to download the supporting data. And then this portal has several different visualizations. So, for example, a visualization of differential expression between 2 genotypes. They've used a volcano plot for this, and there's a supporting table showing all the data. There are also, let's see, I think this one is just a table without a visualization. Yeah. And then differential co-expression this is a figure from their table. And the table, I'm sorry, a figure from their manuscript. The table is sortable and searchable though. Okay, so that's an example of a more complicated portal. And I want to just quickly run through the types of visualizations that are available right now. DK has made this really nice demo portal to show how to configure all of these visualizations. And I'll just click through a couple of them. Manhattan plot, we're all familiar with. So this top part on each of these pages shows you how to configure. In that first page creation interface that I showed you, this is how you fill in all those fields to create a Manhattan plot. And here's what you get. Kind of standard Manhattan plot that's interactive though. You can mouse over the points and see what's there, and a table with the associations. And let's see what other, okay. Yeah, the featured subtables I've talked about. This is a detailed guide on how to set them up, and then what that specifies is these sub rows for each row that you can add supporting evidence or other evidence, and also how to configure the score plot. Volcano plot I showed in the APOL1 portal. This is again details on how to configure it. And finally, also heat map, as I showed before. That's how to configure it, and you can make a nice heat map like this one, and also have the data table. So another visualization that DK is working on, that he alluded to, is this region plot. And this is a kind of a test case right now. It's still under development, but this is a plot that's similar to the locus zoom visualizations that you're probably familiar with in some ways. So it goes across a chromosomal region, and you can see the genes and the associations. In this case, we're looking at T2D associations. And this visualization is drawing on the data in our bioindex. So that is the bottom line meta-analyzed p-values for the associations of all of the data sets in the portal for each phenotype. So that's what we're looking at here. And so,
Maria,
yes. Yes.
there is one question.
Sure.
And the question says, is there a way to zoom in over the Manhattan plot?
Zoom in over the Manhattan plot. DK would be the best person to answer that.
No, not at this point.
Okay, thank you. Sorry to interrupt.
No, that's fine. I mean, you could say that this view is zooming in. You know, it depends on how much you want to zoom in. Okay. So in addition to the region plot, there's an LD plot, which shows you for each of these variants, how many variants and their association significance are in each range of LD with the index variant. And you can click to change the index variant, for example. So we just changed that. There's a table below showing all the data, but then you can also add to this view. So let's add, right now there are only 3 phenotypes. We will be implementing this for even more, but let's add BMI. So that gives us another plot of just like analogous to the T2D plot with BMI and the LD plot. But then the different thing here is that we now also get a third plot that combines the associations for the two. And this can be really useful. So they're color coded, the T2D are blue and the BMI are green. For this particular region, you can see the BMI, if you look at the scales on the individual plots, the BMI associations are much less significant than the T2D associations. So when you combine them, you kind of get this field of green. Whoops. I'm trying to get rid of that box.
Yeah, that is annoying.
Okay. You kind of get this field of green with the T2D associations higher. But now in the LD plot, you can also see the T2D and the BMI associations and their LD relationships. And the same variant in each case is connected by a line. And if you want to change the reference variant, for example, you can set it for either of the phenotypes or both. And you also have a view of what the p-values and betas are for each of the phenotypes. And then, oh yes, when you add a phenotype in the table below, you can see for each variant, again, color-coded, what are the p-values and other parameters for T2D and BMI associations. So I think that is about the end of my demo. I should mention that the DK is also working on a really detailed tutorial that's not quite finished, but the beginning parts of it are ready. And so that's a great way to work through and learn how to use these interfaces. And of course, we're very happy to help you if you start using this and have questions. Just please get in touch and we can help you.
Maria,
So,
before you turn over, can you show everyone where they can find these tutorials on the pages that you built on the UJAMP landing page?
Yes, yes. So the HugeAMP landing page is kp4cd.org. Oops, .org. Okay, that. Yeah.
You found it anyway.
Okay, so this is our, I'm seeing the editing interface because I'm an admin on this site, but here, okay, there's a description of how you can contribute to our effort in many ways. And here, so create a standalone research data page or research data portal. This is the service that we're talking about, the BYOR service. So if you click here, you will see just kind of a landing page for this whole thing. It describes the whole service in general. It gives some links to the evidence. I'm sorry, the examples of its use, and then has a link to the tutorial and also the demonstration portal with all the visualizations that I just showed you, but then also the detailed tutorial. So this is the beginning of the tutorial, and at least the first parts of it are complete.
And our hope is that, if you really like, we don't necessarily expect you to go out and do it yourself, although you can, as Kenny is going to show you. But really, we'd love to collaborate with you, because in each of these cases, the visualizations you see were directly a result of the contributors saying, I need this visualization, let's build it together. And it's made the resource more rich. So it's been actually a really fun experience for us. So, we like to work with you directly, and we're happy to do that, but also, take a spin yourself, the results that you want to make accessible. And I think one of the things we were really hoping for this, and I think Kenny is an excellent demonstration of this, is that we want to help. So many journals require people to make things accessible with publication. And also the types of tables and figures don't necessarily translate well, right? If they're just in a paper. And what we're hoping this service allows is that interactive visualization, easy updating, version control, and accessible to all. So with that, I think I'm hoping it's a good entree into the work Kenny has done to represent his results. Kenny, take it away.
Yeah, thanks, Noel. I think I'll start by just saying, to echoing what Noel just mentioned, which is, I think we came to the Knowledge Portal team saying, hey, we need to host some of these data. We know we want them publicly available. We'd love to have some visualization. And by the way, it's kind of a little more complex than a normal GWAS. There's a GWAS, but there are 20 biomarkers. And by the way, we have this follow-up thing. How can we do all of this? And they said, we have a solution. You're going to do it partially yourself, and that will be better. And it worked out quite well. So I'll give a little overview. Why don't I start here with showing what the actual page looks like, and then I can kind of reverse back towards a little of the echoing some of the stuff Maria mentioned and show the guts of how we got this to happen. So for a real brief, I won't spend a lot of time on this, but just a brief understanding of our project in general. Our goal at a high level was to discover an increasing amount of gene-environment interactions, and sort of spanning across genome-wide analysis, spanning across over 2,000 different exposures. So this was a lot of analysis. And part of the point of this whole project was to take that massive search space and condense to a much smaller set of genetic variants, that if we have a smaller set of genetic variants and a smaller genetic search space, we can use that smaller space and make it feasible to search across thousands of environmental exposures and tens of cardiometabolic biomarkers. So this was basically a two-stage analysis in the UK Biobank. Again, we had a bunch of individuals from multiple ancestries, which were analyzed in a stratified way. We had 20 biomarkers, and we sort of went through, and step one was to conduct what is essentially a GWAS for what we call variance QTLs. I'm not going to really go into that here, but the idea is we conduct what is effectively 20 GWAS with a little twist. Based on the loci that come out of that as significant, we take those genetic loci forward and we perform this exposome-wide interaction analysis, searching for gene-environment interactions. And that ends up producing this two sets of results. One set of variance QTLs, which again, where the results will look like just a basic GWAS. We can visualize with a Manhattan plot. We have a basic set of summary statistics, and then we have gene-environment interaction results. These data are kind of big, so it takes a little bit for this to load here. But ultimately, what we were able to get going on, let's see, again, wait. What we were able to get loaded here is a set of Manhattan plot and tables for each of the particular biomarkers. And so this should be done in just a moment or two. Come on. So anyway, so ultimately what we'll have here is a Manhattan plot, and we'll have a table that show up. So we were able to sort of program this Manhattan plot in. We could go in here, for example, and use this interactivity to say, instead of a p-value of 0.01, we could use a p-value of 0.0001 that will subset. And as you can see, now we've just subsetted this Manhattan plot. So we're looking at a different range here. There are also other ways to subset the data here. We can look at a different biomarker as well, if we wanted to, of the 20. And then finally, I think one of the most important parts here, as simple as it is to be able to, once you're done looking at this, is to be able to then go in and download these raw, not the raw data, but these summary statistics that are exactly what we're trying to make available after having produced a publication describing this. And that would be very easy to both visualize here and to save as a text file for use elsewhere. So that's the VQTL page here. We can then move over to the gene environment interaction results. Right now, this is fairly simple. We're hoping to expand this in the future, but for now, this is essentially just one big table of all the results for these gene environment interactions, where we can give, just like typical summary statistics, we can have a bunch of information about the locus and the SNP that's involved, the outcome, the exposure that was used, and then an effect and a p-value. So, the same types of information that you would expect in a GWAS type summary statistics, but again, easy to visualize, to filter, to download, et cetera. And then the final page here is this research methods page, where we can this is the workflow that you saw on the landing page here, the basic diagram, but then we can also add basically, let's say, a trimmed down versions of a methods section, so that if someone is looking at this page, they can get a little more detail as to what we did and why we did it, so that it provides a bit more context. It's not shoved in your face, but it's very easily accessible here if you want to take a look at that while looking at the page. Okay, so after that high level overview here, let's go back and see a little bit about how this was able to be created. It was astonishingly easy, I would say. So the first thing I'll say, let's actually go to the file manager here. So if we're in this knowledge portal system, we have a couple basic options here. I won't go through all of these, but essentially let's start by saying, okay, we have a bunch of summary statistics from this VQTL analysis, just like GWAS summary statistics. So we could go look at manage your files here. That brings you to a page like this. And essentially the way we chose to do this here is for each of these biomarkers, albumin, ALP, ALT, et cetera, we can just upload a file, a CSV file containing summary statistics. That looks like, actually, I won't show it here because I'm not sharing the right screen. Anyway, it doesn't matter. It's a basic CSV file with summary statistics. And then we can upload that, as well as here, this EWIS meta-analysis. That's the set of gene environment interaction summary statistics. So again, we're basically formatting things in a very straightforward way, the type of way you'd put in a supplementary table or that you'd use for export anywhere else. So given that, we can go, let's see, view your pages, right? So this is the place where you, as the person creating your site, are going to be starting. So first, let's say, okay, we've got our data uploaded. Now I want to just create a landing page. So again, this is this page that I was just showing you. It has an image, it has text, pretty basic. We can come in here, pick a page, give it a title, edit it, and it's going to be pretty straightforward. Because this is a landing page, we don't need a lot of data points or any of this kind of stuff there. We're basically going to go down and we can use this interface here to put text. We can input an image, et cetera. That's going to constitute the landing page. And this is going to be sort of a basic HTML page. So we don't need anything too complex. And then we're simply going to have some node ID where we say a way to reference this landing page if we want to link back to it or something like that. And we can ultimately just save the page, and that's going to produce this home page that you see here. And then to give you an idea, so that would be a landing page, right? Something pretty basic. It's just HTML, it has an image, has text. So now if we get a little more complex and really where I would say maybe the power of this portal comes into play is if we go and we're looking at how we might create, for example, the VQTL results page. And so this is probably the most complex one, still not super complex, but the one that requires the most use of the guts here. So what we're ultimately going to do is say, all right, we want to add some data points. Where are these coming from? They're coming from the file manager where I just showed uploading all these summary statistics. And we give basically a list saying, hey, you want to look for data points, look for them in these files names that we've uploaded. Having uploaded those, okay, let's translate these file names into actual biomarker names. So this file is going to correspond to albumin biomarker. This file is going to correspond to ALP, et cetera. And then there's other stuff like this where we're going to say, all right, well, there's a field called p-value that we want a field called p-value. It's called p in the summary statistics. Okay, things like this sort of basic ability to use a fairly standard format to kind of change names, to maybe paste together a chromosome in a position to create a locus field, basic things like that are very annoying, but commonly necessary things to do with summary statistics. And that this just makes it a little bit more flexible. So now we can provide a couple filters here. So what you saw were the ability to filter by locus, by p-value, or by SNP. These are kind of, again, there's a whole syntax here that I don't know much about, but you can look up if you need and just enter the field you want to search on and how you're going to search on it if you want to subset a table or a Manhattan plot, something like that. And then ultimately, let's see, where did the Manhattan plot go? Aha, here. And this was, a lot of these things are the things that Maria showed in DK's portal, where you see all the inputs that you might want to enter. And this is where you're going to enter all of these things. We can say, okay, I want a Manhattan plot. And again, this is what you looked at before as an interactive Manhattan plot. That was as simple as going above and specifying what each of the fields mean and where to look for the data. Say we want a Manhattan plot and just say, okay, the x-axis field is going to be the locus tab. The y-axis field, we even have a little bit of ability to make conversions here. So we put in a p-value, we're going to specify it in the syntax here such that we get out a negative log p-value, et cetera. And this is a pretty smart system, so it understands how to convert these things like a p-value to what is necessary for a Manhattan plot. And that's pretty straightforward. Then again, we can put in other things. We can put in an HTML text just like we otherwise could, some basic description of what you're seeing in the plot. And then again, there are other things that we can reference as well in this table. So I mentioned before that we have a node ID. So each of these pages has a unique way to reference it. And that's really useful because then we can go back and say, all right, we're going to create a methods page. That's what I showed with the workflow diagram and all the methods. Okay, great. We can reference that page. And so if we want, I'm here on the, let's say the GBYOR results page. I want to be able to go from that page and say, okay, what are the research methods associated with that? How do I know, how does the site know where to go here? Well, we can use these unique node IDs and kind of go back and forth. So I am probably getting far too into the weeds here because I'm excited about it. But anyway, so this hopefully gives an idea of the ways that we're using kind of a little bit of HTML, a little bit of just manual typing, a little bit of data munging to create the kind of seamless visualization that you see on that main page. So I think that's the main sort of thing I wanted to share there. In general, once again, just to echo what Noelle mentioned, I think this is really exciting for me personally, because I think this is kind of like the paper of the future in a sense, right? I mean, this has methods, it has the ability to show results, to show graphs, but also to, with one click, download those summary statistics or use them somewhere. In contrast to a static paper where it's really annoying to go get any of the data or understand additional information. So I am a huge fan of being able to interact with the results from a paper and understand them in an interactive way. So yeah, big thanks to the CMDKP team, especially DK, for all the help with getting this up and started. And also to Joanne Cole, she is another big contributor to this project. She's also at Broad and was helping to supervise a lot of this research. So big thanks to Joanne. And yeah, I will shut up now and happy to talk any further with questions or anything else.
Thank you, Candy. That was awesome. I learned something too. So that was great. We have a couple questions. One, I think it's already been answered, so it's actually good to know. Do we need an account to get started? DK, do you want to answer that live?
Sure. So which question do you mean? The converting p-value to minus log 10? Yeah, it's built in. So you just need to indicate, okay, I want to convert the data into minus log 10, and then it will do it. So it has some data conversion capabilities that you can join and then calculate or generate scores based on the values in other columns. So the score now, as I mentioned in the beginning, it's in beta stage and it's getting more functionalities. But yeah, so minus log 10 is built in, and we will have no more in the future.
Thanks, DK. And then another question was, do you need an account to get started? And Maria answered yes, but the details are easy. Anyone can get an account, right, Maria?
Yeah.
You just need to kind of register, just more so we can give you access to it. But it's on there already in the tutorial and teach you how to do it there. Any other questions?
Yeah, there's another one in the Q&A. Yeah. Can the figures built through the portal be used for publication or project submission?
The answer to that is yes.
Depends on, if you, so it doesn't, okay, so ideally, if you just insert a link, then it can build figures in other places. That would be ideal, but it's not possible yet. But you can build figures and then screen capture and insert in your paper. Yeah.
But for project submission, I can imagine that would be already something that we could say that we can do. Specifically, ideally, this was really important in many cases for people because the journal would not publish without this being a portal for a couple of these instances, which is why we actually really pursued this. So the journal said you need to make these results accessible in order, like the full results. And rather than put them through our entire pipeline, which would take time and is not necessarily necessary, we built this to do that. So it does serve the publication requirements for many journals in terms of accessibility, which is, I think, one of the major reasons why we did this. Well, there's quick questions. Since there's still time, I will mention one other portal that we built, which I think is really valuable to share. Let me share my screen real quick and take you there. The reason I think this is a little bit beyond the scope of what we talked about today, but it gives you a sense of where we also want to head, because it's not just about building pages, but also representing different types of data. And we did this with collaboration with, there you go, Bob Fries and Toby Walter, to build a portal for an organelle that is underrepresented in their mind, called the Lipid Droplet Knowledge Portal. And this is also a subject of, it's on my archive now, but we also just resubmitted it for publication and along with the results. And what was nice about this is that they themselves have a very small community of people who work on this organelle, and inside their own community, they wanted a way to share all of their results and data. And so we built this a couple of years ago, but we've been developing it along the way to expand this. And this is really quite elegant in the sense it just doesn't, it's beyond human data, it has fly data, mouse data, but it has elegant visualizations that represent the results from that specific data set, including images, which is really nice. So this is a full collaboration, this is a full stack that we've built, but this gives you an example of the other types of expansion and directions that we also are interested in going. This is a little bit not build your own, but this is a bit more collaborate with us to bring your own results. This is all about results. Again, what's nice about this is it also leverages some of the human genetics data that we have in the other resource. So you can also take a tour of this portal. It's open access. It allows you to do several different types of visualizations, which we didn't even show today. So it's worth noting. And you can find it on the same exact page that we referenced earlier. And moreover, one of the things that we touched upon along the way, and you saw in the region plot that Maria showed, is, a lot of the use cases I'm hearing are, I want to see my results with some data that I don't have. And I think that's another sort of development avenue that we're pursuing, which is, see association results across a different set of traits, but also layer in annotations from tissues and cell types that are relevant to my trait that I don't have access, that I don't have aggregated. Well, we do, and we can make those accessible to these different portals in the way that they might like in the concert of their results. So that's another, that's the leverage that I was mentioning. So with that, any other questions? I'll stop sharing. Again, you can find this on kp4cd.org. You can also find it through our main page, like Maria just said, which is great. She's reading my mind. We hope to reach out to us if you have any questions or want to work with us or have some ideas to make it better. I think that would be really valuable to us. And I think, unless we'll end early, give people 10 minutes back, because I imagine that's really necessary at the end of the year, given that we're all trying to do lots of things. In terms of announcements, our next webinar will be February 10th, same time. And I think what we're hoping to do for that is give you sort of a roadmap, and I invite Jason to come talk about the vision for the future of the huge AMP research and the Common Metabolic Disease Knowledge Portal, and as we expand into broader common diseases. If you have any ideas for webinars as well, please shoot me an email or anyone on this, and we'd be happy to add them as well. So I'll thank Kenny for joining. That was really helpful. Thank you, appreciate it. And Maria and DK for the vision, making it, Maria helping it turn into a product. I think this has been a really fun experience for me too. So have a great new year, everybody, and we'll see you in 2022. Thank you.
Thanks, everyone. Bye-bye.
Thank you.
Bye.